So on Pavlovs Maggots. And so thank you for coming. I tried to be as simple as possible in my language because I'm doing neurobiology. I don't know how many neurobiologists there are here. And yeah, you are. So the question that my interest is on sensory discrimination. And why sensory discrimination? Sensory discrimination is a universal challenge for all animals. So from flies to humans, brains need to discriminate sensory information, form representations of sensory stimuli in the brain, and this sensory stimuli is integrated, stored as memory, and this is actually it's associated with other sensory stimuli, with contextual cues like punishment or reward, and this is what is stored in our memory. So if you think about the environment here, there are many molecules, there are many photons. How does the brain cope with this amount of information? And what it happens in the brain is that humans, for example, can discriminate like 1 trillion odors. And you can discriminate the smell of a banana or a strawberry. A banana itself has 250 chemicals. And also we can...
How do we know that we can discriminate against 1 trillion odors?
Well, you can't really test it. It's theoretical, actually.
Okay. It's a long experiment.
And we can discriminate sensory objects, like a piece of music, or the face of a celebrity. And what we know is that neurons in the higher brain become more and more selective. So for example, olfaction. In olfaction there are many chemicals, organic chemicals. These organic chemicals, they have functional groups, the carbon chains have different lengths, and what we know is that the receptors in the epithelium here, they recognize these epitopes, these species within these molecules. However, we don't know exactly how this happens. And however, the sensory neurons, what we call them, they are very specific. They react to specific bits of the molecule. And so the first step in the olfactory system is analytical. You have a molecule is decomposed, sort in pieces, and this happens in the first center, in the brain, in the olfactory bulb. And then after that, in the higher brain, in the association cortex, all these channels, let's say, channels get integrated and they form a whole perception, like a banana or a strawberry. So we don't know how this happens, and I am interested in these mechanisms. So I have chosen to study the maggots from Drosophila because these maggots are very useful and there are 10,000 neurons in one brain hemisphere, and we have 20 billion neurons. So they are manageable, we have less neurons, the system is simpler. And also I can use the very powerful genetic tools that we can apply to flies in Drosophila. So if you want to understand the mechanisms, we have to isolate the neurons. We need ways in which we can block these neurons and see what happens in the behavior or what happens in the activity of these neurons. And to do this, we have very good tools in Drosophila that we can apply in the maggots. So I'm going to describe just one of these techniques because this is the basis to understand all the genetics that we do in Drosophila. And you can see here a male is flapping the wing because it is a very stereotypic behavior, a courtship behavior, and this is the female. So we can generate flies that has a protein called GAL4, and this protein is, we can have it in different parts in the genome. And when you have this protein in different parts of the genome, it means that it will be produced depending on a spatial and temporal pattern. And this means that you can label with this technique different neurons. So we have on the one side these GAL4 lines. There are databases and there are stocks around the world. A few centers have tens of these stalks. And then you cross these ones to other flies that will have a reporter gene. And when girl 4 gets together with a sequence in front of this reporter gene, will drive the expression of the reporter. So we have on the one side set of lines that will drive the expression of these proteins in a specific pattern, and on the other hand we have these lines that will report the expression of this protein, for example GFP from the jellyfish, and this GFP will give fluorescence, and this is how we can mark the neurons. Or you can express toxins, and by expressing toxins you can block neurons, and some other things that I am doing, for example, that is you can express proteins that bind to calcium, and then using this in combination with GFP I can record neuronal activity. These lines are very useful, but they are very complex. It's not always easy to have just one neuron, but we have other techniques to see just one neuron. But this is the most basic technique that we use. And one important thing is that we have these GAL4 lines, we call them drivers of gene expression, and we have also other type of drivers. And so we can manipulate the expression of one protein with one driver and another protein with a different driver. And I will show you during my talk what we can learn from that. So this technique is, we can use it in the fly and we can use it in the larvae. And the maggots are able to discriminate odor. And we know that because of Pavlovian learning, olfactory learning. So this is a result of a choice test. You can put a maggot on an agar dish, and you can give a smell and an electric shock at the same time. Then the maggot will remember that that smell is associated with electric shock. You can also put a maggot on agar and have sugar there together with the smell. And so the maggot will know that that smell is associated with food, reward. So they are very small, and you might think they're not very intelligent, but they can discriminate odors. So this is a result of a choice test. And what you see here, for example, these maggots are migrating away from octanol there. And so they're migrating away from octanol, and they go to amyl acetate because octanol was paired with electric shock. On the other hand here, maggots are migrated away from amyl acetate because amyl acetate was paired with electric shock, so they go away. So from this Pavlovian discrimination test, we know that larvae are able to discriminate octanol from amyl acetate. And then where does this associative learning happens in the brain? And this happens in the mushroom bodies. And these mushroom bodies are associative learning centers in the insect brain, and it is also present across many invertebrate phyla. And we know that because there are mutants, structural mutants that are defective in this structure, and when you do this test of factory learning, they are unable to learn. You can delete these structures from the brain during development. You can block the development, and they are unable to learn as well. And then using more genetics, we know that many proteins that are involved in learning are actually expressed in these mushroom bodies. And so if all these mutants of learning, if you re-express the normal protein, you can rescue. This is a bit a genetic trick. But the main message is that we know that the mushroom bodies are centers of associative learning. So what is then the circuitry of associative learning? And the circuitry is, we have here, this is in the case of insects, the antennal lobe structure there. In yellow, you see the glomeruli. These are units. So a single class of olfactory receptors go to one glomerulus. So glomerulus represent one odor quality, one specificity. And then we have the projection neurons. The projection neurons bring information from these glomeruli. For example, the yellow one, if you follow the red line, and bring this information here into the calyx of the mushroom bodies. And here, this is the input, the sensory input region of the mushroom bodies. Here you have the mushroom body neurons called canyon cells. And memory forms when there is coincident activation of these canyon cells together with punishment signal or reward signal. So this is the model that we have at the moment on associative learning. And my interest is here in the calyx and how these sensory stimuli are discriminated and what is the circuitry for it. So the questions that I am addressing is how are these odor specificities? I call odor specificities to the ligand or to the bit that makes that activates a specific sensory neuron. How is it represented in the brain? And what are the other novel neurons that are in the circuitry, and how these neurons modulate the activity of this center, and what is the role of inhibition in this center. So this is a third insta larva. So there is first, second, and third insta larvae. To give you an idea, this is about 4 or 5 days after egg laying, and the generation time of the fly is 10 days, so it's quite quick. So it's a very good biological preparation to use. And so this is the mushroom bodies. You see here the canyon cells, the cell bodies. This is the dendritic area, the calyx, and these are the pedunculus and the lobes. So olfactory information, the sensory neurons, these are the sensory neurons that bind the ligands in the nose. They carry the olfactory information here into the antennal lobe. This is the first olfactory center. And as I said before, this is the part that is analytical. There are parallel channels bringing distinct specificities into the antennal lobe. And from here, projection neurons bring this information into the calyx of the mushroom bodies. And the calyx of the mushroom bodies process this sensory information. And then here, pedunculus and medial and vertical lobe, these are regions, these are output regions of the mushroom bodies. So far, okay. You have any questions? Please let me know. So this is how the mushroom bodies look like. And so using a genetic technique, we can label single neurons. And these are single neurons, are projection neurons, are the neurons that connect the first center antennal lobe to the calyx. And by generating these single neurons, we generated multiple cables that connect the first center to the second center. So, for example, this is antennal lobe, and you see, for example, here, well, maybe here, it's better, it's closer. Here, you see the antennal lobe, the first olfactory center. And here you see, in green, you see the process of one of these neurons, the projection neurons. So one process innervates here, in this part. If you see in different individuals, you see the same green at the same position. And then this neuron innervates the antennal lobe, and then it innervates the calyx. And in the calyx, in all 3 individuals, it innervates this part. So what it means is that the antennal lobe is a structure that has 21 units. Each of the units represents one specificity. It's the input of one sensory neuron. And by doing this connectivity between these units of the antennal lobe and units in the calyx, we can make a sensory map of the calyx. So by doing this, I made distinct cables, and I went, okay, from this from this receptor 1A, I went into the brain and I mapped the 1A in the calyx. And this is what I came with. So these are in color coding. You see all the inputs from one sensory neuron through the projection neurons. These are actually inputs from the projection neurons, from the cables that I show you, but they originate from one specific sensory neuron. So what we have here is that there are 21 antennal lobe glomeruli. We call these units glomeruli, but remember that are units of specificity. And there are these 21 glomeruli connect to 23 glomeruli in the calyx. So it's parallel channels. And interestingly, anatomically, I marked previously 34 of these units. So there is olfactory input, the color-coded one, but there are other inputs here that we don't know. And interestingly, there is also another input here of a neuron that comes from the SOG, which is a taste center. So this predicts that in this area of the brain, they are integrating different information modalities. So I told you the cable properties. I made a sensory map in the mushroom bodies. So now the question is, how do the neurons in the mushroom body integrate all this information? And if you look, if I look carefully at one mushroom body neuron, which is the canyon cell, you can see here the cell body, and you can see that it innervates about 6 of these units that are labeled here. And these are in dots. So this we call glomeruli. And here you can see, for example, the terminal of one of these glomerulus. And it is a claw-like structure around one of these units. So what it tells is that this canyon cell is integrating, might be integrating the information coming from these 6 different units. And this is an anatomical study. And this is actually what happens with the activity in the brain physiologically. By following the cable properties, we know that one sensory neuron connects to one antennal lobe glomerulus. One antennal lobe glomerulus is connected to one calyx glomerulus, mostly. So it's the same in terms of activity. And what you see here is an image of 2 calyces, the 2 brain hemispheres. Have you seen 2 glowing glomerulus? So this is a live larva, and I give a puff of odor. And in this larvae, there is only one sensory neuron that is functional. Okay? So genetically, I can delete all the sensory neurons and just target one sensory neuron and rescue the activity in one sensory neuron. So this larva has only one sensory neuron functional. And from this sensory neuron, I can see, if I give a puff of odor, that the activity is localized mostly in one glomerulus. I have seen examples of 2 or 3 glomeruli. There is some spread of activity, but it's a very localized, it's mostly localized activity. So my results tells that in the periphery you have many neurons bringing different channels, and the canyon cells are integrators of these parallel channels. And this anatomical organization is consistent with electrophysiological results from another laboratory, the Churchill laboratory. And you see the projection neurons, the immediate neurons that bring the olfactory information into the mushroom bodies. So the projection neurons, they respond, this is a panel of different odors. And what you see here is the spikes of these projection neurons. And projection neurons respond if you give, we work with pure odors, but if you give 12 odors, they're going to respond to these 12 odors. And the responses are also long-lasting. So they are broadly responsive. They respond to many odors. They don't have much selectivity. But if you look at the mushroom body neurons, the canyon cells, they respond only to few odors, you see here. And it's very transient, the response. So there is a dramatic change in the coding, in the representation of smell between the peripheral projection neurons and the mushroom body neurons. So this is consistent with the anatomy that I see. And so this is a summary. And if you follow just one of the color, this is olfactory sensory neurons there in the nose. they will detect one odor specificity, and they bring it here to the antennal lobe, to one of these units called glomerulus, and there is a one-to-one connectivity there. Projection neurons connect these antennal lobe glomerulus to the calyx glomerulus. And you see that these are all parallel channels. There are 21 parallel channels. And canyon cells, on the other hand, they have these 6 branches, so they receive different inputs. The 6 branches innervate different glomeruli. So they have 6 branches, and apparently they don't have any stereotypic, and they are randomly connected. And so we have here 3 canyon cells, and they show this pattern. So they are integrators. They seem to be integrators of multiple inputs. So the next question that I am addressing is how the canyon cells integrate all these multiple channels and make the responses specific, selective. And here again, I use the flight genetics to screen, to identify Galfo lines that would mark neurons in the mushroom bodies, and specifically in the mushroom body calyx. So for example, these are 3 lines, and this is in red, you see the glomeruli, and this is the calyx region. And you can see in blue is GABA, the inhibitory neurotransmitter GABA. And in green, you see these novel neurons that I identified. So, for example, in this one there is an overlap between the GABA, the inhibitory neurotransmitter, and the neuron. In this one here, you see this green here inside in the core of the calyx. This is another neuron, and this also is another neuron, and some of the terminals of this neuron labeled in GFP are in close contact with GABA terminal. So I had now a panel of Galfo lines that label different neurons, novel neurons in this center. And I focused my attention on this one there because it's an inhibitory neuron, and I went into analyzing how this inhibitory neuron regulates the activity in the calyx. And if you can cross your eyes, you can see a very beautiful 3D there. I don't know who is able to do that. Have you seen these 3D pictures that? Yeah. Is someone able to see the neurons here?
So just by crossing our eyes?
Yes.
To try to merge the 2 images. When you cross your
Do you have to be quite close?
Not necessarily.
Cross your eyes. Try to merge the 2 images into one when you cross your eyes.
Yeah, got it now.
You got it. So.
It's because you've got an arrow on one of them and not on the other. I get looking for the other one on the right hand side.
So this is the cell body of the neuron, this one there, the cell body, and it has a branch there, one primary process, and this bifurcates into here, the calyx of the mushroom bodies, and the other one goes here into the lobes. So in green you see the neuron, in red is the neurotransmitter, and in blue you can see the mushroom bodies. So we found this neuron that is a GABAergic neuron. Someone else can see? No. We can try later. So this is a GABAergic neuron, and it's the only GABAergic neuron in the calyx. And because here in red you see the GABA accumulations, the GABA buttons we call them, the terminals, and all the GFP, all the GABA buttons overlap with the GFP of the neurons.
So what is GABAergic again, sorry?
Oh, GABA is an inhibitory neurotransmitter.
Yeah.
So the brain is a balance between excitation and inhibition. Of course, there is also modulation. So the prediction here is that if you have excitation by receiving odours, there must be some mechanisms to, if the neurons are highly selective, you would expect to have some kind of inhibition in the system. So GABA is a neurotransmitter that is released by the inhibitory neurons and will depress the activity of the neurons. Yeah? So,
and then GABAergic is just a name for the neuron that does this.
yes,
Okay.
and so not to confuse, we call it EPL. We call it EPL. Okay? So we have now the EPL neuron, which is an inhibitory neuron. So how we go about it? So it's the only neuron, inhibitory neuron, only GABAergic and only inhibitory neuron that we have in the calyx. And for example, this is another neurotransmitter that is octopamine, which is actually the equivalent of noradrenaline. And if you see here, for example, the calyx in blue and octopamine in red, and here are the EPL neuron, you see, for example, that it doesn't have octopamine. So this neuron does not possess, does not produce octopamine. And another thing that we can do, neurons are polarized. And it means it has processes that receive signals, and it has processes that deliver the signal. So, for example, the Kenyan cell receives signals in the calyx, and the output is the lobes. And concerning the EPL, we can analyze which are the processes that receive the signal and which are the processes that deliver the signal. And we can do that by expressing proteins that that label what we call the presynaptic terminals, which are the sites that receive signals, opposed to postsynaptic sites that are the sites that release the signals. And for example, here, this is the lobe region, the output region of the mushroom bodies, and you see here the red, and this is labeled by a protein that accumulates postsynaptically. So what we know from this experiment there is that they are receiving signals. The EPL receives signals in the lobes. On the other hand, in the calyx here you see the green, and this is a presynaptic marker. So we know that in the calyx this neuron must have terminals that is releasing signal. So we can learn how this neuron is connected in the mushroom body, where it receives the signals from and where it brings the signals to. But this is a very rough experiment. We can only know where the terminals are, where they receive and where they release the signal, but we don't know which neurons is connected. And this we can do is another trick. We can express GFP. GFP, you can make two parts of GFP. If you split the GFP, it's called split GFP actually, you don't see any fluorescence. But when the GFP comes together, you see the fluorescence. So you can express one part of GFP in one class of neurons and another part of GFP in another class of neurons. And when these neurons come together, you will see GFP. And by come together, I mean in close proximity. And so they are suggestive of synapses, but they are not evidence of real synapses. But they are very close together, as close enough to be able to make synapses. And this is what this is one split GFP experiment between the mushroom body neurons, the Kenyan cells that receive signals, and EPL, the inhibitory neuron. And here you see the GFP here in the calyx, and you see GFP here in the lobes. So we know that EPL is probably making contact with the mushroom body in the calyx area and here in the lobes. And here is the buttons in red. I will show you a higher magnification of that. There you see the calyx, a larger magnification. You see the glomeruli in blue, and in red you see the GABA terminals. These GABA terminals are surrounded by GFP, which is EPL processes. And these are the sites of contact between the APL and the canyon cells, because you only see GFP when the two cells come in proximity. And so we know from there that canyon cells, the mushroom body neurons, are making extensive contacts with APL. And here you see some GFP dots, some split GFP signal, and this is with the input neuron, the projection neurons that bring olfactory signals. And these projection neurons seem to have some contact with the canyon cells. So this is a split GFP between the canyon cells and the input neurons. So they might be also regulating some of the inputs. And this is how this inhibitory neuron looks in the calyx, and a larger magnification. You see in red the GABA termini, this is the inhibitory neurotransmitter, and you see here in green the processes of the APL. And if you look carefully, there are very large buttons in the periphery between glomeruli, and in the core of the calyx you have smaller buttons. And if you give an odor to this neuron, this is again the larval brain, and you see here the buttons, large buttons. So here it comes. They respond to smell. So this is the head of the larvae. I'm giving a smell and I'm recording from a single neuron in the brain. And also you see activity in the whole calyx, because as I show you, there are little buttons everywhere in the calyx. So this APL responds to multiple odors. It's not specific for one odor, just responds to any odor that you give it. So how does this neuron work? In order to understand how this neuron works, I mean, which neurons activate this the APL, we have to do some tricks. For example, block neurons and record activity. And the way we can do that, as I show you in USQL4, we can express proteins, for example, that at lower temperature, they work normally, but if you raise the temperature, they don't work anymore. Proteins that have a mutation, and the one that I am using here is called Chivire, is a dynamic mutant, which is a protein involved in vesicle recycling. So I can't block the recycling of synaptic vesicles. So this is a way of stopping synaptic transmission. So by expressing this protein in mushroom body neurons, I can block the output of the mushroom bodies. So this is just a graph. But what I'm showing here is that if I block the output of the mushroom body neurons, APL does not respond anymore. And this is in the Y you see the responses of the APL, and here I see the temperature. So if I go to higher temperature, the protein doesn't work anymore, then there's no more recycling going on, there's no more synaptic transmission, and there is a gradual depletion of synaptic vesicles. But if I go back to the normal temperature, to the lower temperature, then it goes up again, and synaptic transmission is restored. So it looks that APL is receiving signals from the canyon cells. So the model is that, as I told you, there are parallel channels, 21 parallel channels that bring olfactory specificities into the antennal lobe. Projection neurons connect one-to-one here and bring these parallel channels into the calyx. There are canyon cells here, the mushroom body neurons, that seem to integrate all this information. And then I have now this inhibitory neuron that is receiving the signal from the mushroom body neurons and inhibiting the activity in the calyx. So the conclusion is that at the moment, I think that this larval APL is important in regulating the activity of the mushroom bodies by this inhibitory activity. And I show you that this neuron anatomically connects from canyon cell output lobes into the calyx. And I show you that if I blocked the canyon cell, the output from the mushroom bodies, this neuron is not active anymore. So it must come, the activity must come from the canyon cells. So what we are doing next, APM neuron is connected with canyon cells, but it might be connected to other neurons as well, and it might be regulating input activity. We don't know that yet. And I have also, we only know about canyon cells, the input projection neurons, and these inhibitory neurons. But there are other neurons that we found that are modulatory neurons, and we are now investigating how the activity of the mushroom body is regulated by these other neurons, which can bring many signals, like sleep signals, attention signals, the physiological state of the organism, etc. And this is a very important slide because I started this work many years ago when I was in Japan. And I want to thank my collaborators because they have really been very, very, very helpful. And Keito and Takeshi Yabasaki, for this work, they provided images from yarful lines, from larval brains, yarful lines that I talked to you about. And there are, I would say, a few places in the world that they keep these yarful lines, and they gave me images that I could screen, and they have been useful in identifying new lines. And Raini Stoker and Nanai Chandru, they are the ones who we had a really nice collaboration. They made all the cables from antenna lobe to calyx, and this is a lot of work to do, and I was able to analyze the connectivity thanks to these collaborators. And Kaharu Ken, who is here, he's supporting me, and he's participating in the genetics of this project. And I am very thankful to many students, and I had an exciting time with students here in Cambridge. And I want to mention Hilary Wong, Dixie Shah, and Shorsha Salula, and they came as part 2 students to the lab, and they generated 250 yarful lines in just 8 weeks, okay? And then Angela Wang came for a summer, Maria Pepa, Lorna Cox is from Bath, and they analyzed these lines. You know, it's a lot of work. You have to dissect, the brain, I should have brought the brain. The brain, I don't know, it's probably not even a millimeter, It's very hard to work with these things. So the scale of this, I am a large fixture, but the canyon cell body is 5 microns. So this is the scale we are working with. So dissecting all these brains is a lot of work, and they have done all the immunolabeling and looking at the neurons. And I haven't talked yet, and I hope to be able to talk someday. Giles Harper Donnelly and Alex McLachlan have been working in the lab developing odor discrimination assays with larvae, so putting larvae on agar and see whether they could develop a new way of testing odor discrimination. Because in this way I could test these novel neurons actually behaviorally in the discrimination. And I think I don't forget anyone here. And so Angela Wan and Hilary Wong, we are now in the lab, and they are characterizing these novel extrinsic neurons. Thank you.